Students, if you are confused about the inhibition process, this video will explain the entire inhibition process step by step in an easy way and in a simple conceptual exam-oriented manner. So let's start our today's topics of today's lectures about the inhibition process. As you know, there are the various types of inhibition in case of enzyme. So first of all, if we have to explain, there is a inhibition that is called the competitive inhibitions. What is the meaning of competitive inhibitions? Actually, competitive inhibition are also called the substrate analog inhibition. It is a reversible inhibition of the enzyme, and here the inhibitor binds to the enzyme by weak and easily dissociate bonds. So the inhibitor can be removed from the enzyme by the dialysis to restore the enzyme activity. So this is the definition of the enzyme in competitive inhibitions, and in that cases, the some important characteristics features of the competitive inhibition are given below. So first of all, the inhibitor is closely structural similarity and the substrate and the competes with the latter for the substrate binding sites of the enzyme. So this is the first important criteria. And here in inhibitor molecule are competing with the normal substrate. E plus S so on ES, that is E plus P. If we instead of substrate, if we add their inhibitor, then E plus I is equal to EI. Since EI or inhibitor enzyme inhibitor complex can react only to reform the enzyme and inhibitor, the number of enzyme molecules available in ES formation is reduced. Suppose if you see 100 molecules of substrate and 100 molecule of inhibitor are competing for 100 molecule of enzyme. So half of the enzyme molecule are trapped by the inhibitor and only half of the molecule are available for the catalysis to form the product. So number 3, since the effective concentration of the enzyme is reduced, the reaction velocity is decreased. And number 4, what is this? In competitive inhibition, the inhibitor will structurally analog to the substrate, and there will be similarity of in 3-dimensional structures between the substrate as well as the inhibitor. For example, succinate dehydrogenase reaction is inhibited by the malonate. And competitive inhibition is usually reversible, and excessive substrate abolizes the inhibitions. And another important criteria is that from if you see the graph 5.20, that the graph, this is the graph where the inhibitor when binds, then that is called the substrate with the inhibitor, then the curve shows the declining straight line. And whereas the if you have to without inhibitor where the substrate is binding, then the curve is hyperbolic types. So in this way, from this graph, it is obvious to in case of competitive inhibitions, the Km is increased here in presence of competitive inhibitors. Thus, the competitive inhibitor apparently increases in the Km, and in other words, the affinity of the enzyme towards the substrate is apparently decreased in presence of inhibitor. And Vmax is not changed. So the Vmax is not changed in case of the competitive inhibition. So this is all about the competitive inhibition. And if you see the Michaelis-Menten reactions and also inhibitions, there should be Vmax unchanged, but in case of Km is also changed. Another important inhibitor is called the non-competitive inhibitor. And what is this? A variety of poisons such as the iodoacetate, heavy metal ions, lead, mercury, and oxidizing agents acts as a irreversible non-competitive inhibition. And these are the example of the non-competitive inhibitors. And there is no competition between the substrate and inhibitor. And here the inhibitor usually binds to the different domain of the enzyme and other than the substrate binding sites. Since these inhibitors have no structural resemblance to the substrate and increase in the substrate concentration generally does not relieve this inhibition. So this saturation curve in this type of shown in the figure number, if you have to see 5.23, then it will be clearly you can understand how this inhibitor is behaving. This is like this thing. And here double reciprocal plot reactions, and if you see this non-competitive inhibition when the this is called the with inhibitors, and this is called the without inhibitor in case of non-competitive inhibitions. But another type of inhibition is called the uncompetitive inhibition. That is here the inhibitor does not have any affinity for the free enzyme. So inhibitor binds to the enzyme substrate complex but not to the free enzyme. In such cases, both Vmax and Km are decreased. So inhibition of placental alkaline phosphatase by the phenylalanine is a. So student, if you compare in between the competitive inhibition and non-competitive inhibitions, what are these things? This is acting on where it act. Competitive inhibitors, they can bind active sites, but non-competitive inhibitor may or may not act on the active sites. The structure of the inhibitor substrate analog to the competitive inhibition unrelated to the molecules. Inhibition is reversible, generally irreversible. Excess substrate, that is the inhibition relieved, relieved and also no effect. Km is increased, no change. Vmax, Vmax also competitive inhibition, no change, but in case of non-competitive inhibition, that is decreased. And significant drug action and toxicological actions. So this is all about the competitive, uncompetitive, and non-competitive inhibition. And so another important inhibitions we want to highlight here, suicide inhibitions. What are these things? Actually, it is a special type of irreversible inhibition of enzyme activity. It's also known as mechanism-based inactivation. So the inhibitor makes use of the enzyme own reaction mechanism to inactivate it. In suicide inhibitions, the structural analog is converted to a more effective inhibitor with the help of the enzyme to be inhibited. The substrate like the compound initially binds with the enzyme and first few steps of the pathway are catalyzed. This new product irreversibly bind to the enzyme and inhibits the further reactions. For example, ODC or ornithine decarboxylase catalyze the conversion of ornithine to that substance and which is necessary for the polyamine synthesis. So this is one important type of, and another important similar mechanism which we have seen, that is the allopurinol, that is which is oxidized by the xanthine oxidase to alloxanthine, and that is strong inhibitor of the xanthine oxidase. So apart from this, there are another very few types of inhibitors are present, and that we will discuss in like the allosteric inhibitions or feedback inhibition, that we will discuss in our next lectures. But today we are concentrated about this type of these important three types of inhibition, like the competitive, uncompetitive, and even also non-competitive inhibition. I think students, you all have understand clearly this lecture, and so thank you for watching this lecture. Hope this session will help you understand the topic clearly. And if you found this video useful, please like, share, and subscribe to our Physio-Pulse channels. But do not forget to press the notification bell so you never miss our upcoming lectures like the physiology, biochemistry, biology, even also the allied subjects like the biological. And if you have any question, any query, any suggestion, please give some comments in our comment section. Thank you, and we'll see you in the next lecture. Until then, keep learning and stay curious.